Assalamu alaikum everyone, welcome to the Biochem. Today we will discuss about the lipid and the biomembrane structure. So biomembrane is called also the cell membrane. It's a selectively permeable. Selectively permeable means it can transport a particular type of molecule into the cell. This cell membrane gives the structural stability of a cell. These separate the interior of a cell from the external environment or create intracellular compartment by the serving as a boundary between the one part of the cell and another part. So the bilayer structure of biomembrane, there is a two part we can see that one is the head group and other is the tail group. The head group is the hydrophilic group and the tail group is the hydrophobic group. The head group or polar head group protect the hydrophobic tail from the water molecule or other polar or non-polar molecules. So this is how a biomembrane structure is formed. So the hydrophobic effect and the van der Waals interaction between the fatty acyl tail drive the assembly of the bilayer. So there is three types of membrane lipid. These are phosphoglyceride, sphingolipid, and cholesterol. So phosphoglyceride has different type of head group. These are phosphatidylethanamine, phosphatidylcholine, phosphatidylserine, phosphatidylinositol. So different type of amino acid make a difference between this head group. So if we see the sphingolipid, these are also have a different head groups, that is sphingomyelin and glucosylceramide. So these also give a different type of sphingolipid. And the last one is the cholesterol. So lipid bilayer movement, there is three type of movement: lateral movement, flipflop movement, and rotational movement. Lateral movement denoted as the movement of the lipid or protein in the same layer laterally. So if we see the animation, we can see that the molecule or phospholipid are laterally move in the same plane or same layer. This increases the fluidity of the cell membrane. So this is denoted as the lateral movement. If we say about the flip-flop movement, this means when the phospholipid migrated occur from the inner or the cytoplasmic side of the bilayer to the outer or the cytoplasmic side of the bilayer is denoted as the flop, and when it occur from the opposite direction, this is called the flip. So this movement is called the flip-flop movement. So we can, if we see the animation, we can see that this is called the flip, and if this rotate again into this position, this is called as a flop. So this movement denoted as the flip-flop movement. So when we see about the rotational movement, this means the phospholipid rotates on its axis to interact with its immediate neighbors. This type of rotational movement, we can see that this rotate in the same axis, and this denoted as the rotational movement. So here is the experiment of the lateral movement of a biomembrane. So this experiment is also known as the Frap experiments because it named the fluorescence recovery after the photobleaching. We will see this experiment in just a moment. So here is the Frap experiment. Suppose this is a cell membrane. So firstly, or the first step is to label the cell membrane with a fluorescent reagent. So after the labeling the fluorescent reagent, we will bleach the cell membrane with a laser. So here we can see that after bleaching the cell membrane with the laser, there will be few area where they don't carry the fluorescent reagent anymore. So after few moment, we can see that the fluorescent recover occur, and these are migrated to the other places because of the lateral movement. The Frap experiment, we can see that The 50% of the fluorescence return to the bleached area. This indicates that 50% of the receptor molecule in the eliminated membrane path were mobile and the 50% were immobile. This is because of the proportional or the lateral movement of the bleach area in the lipid bilayer. So this proves that the cell membrane has a lateral movement. So this all for today. Thank you for watching.